Proteins can be defined as macromolecules that are made up of amino acids linked together by peptide bonds to create lengthy chains. These large chains fall into two or more globular clusters to form a domain. Protein domains are the fundamental units of protein structure, folding, function, evolution, and design. They are able to perform a variety of functions such as binding to specific molecules, catalyzing chemical reactions, or participating in protein-protein interaction.
The first protein domain is the rhodopsin's alpha helical transmembrane domains. It belongs to the G-protein coupled receptor superfamily, and it uses G-protein activation to modulate signaling and intracellular activities. This part is the alpha helical transmembrane protein classification. It can be classified by the number of transmembrane helices and by the helix architecture. A few protein analysis is used to detect alpha helical transmembrane domains, which are the gel electrophoresis mobility shift assay (EMSA), which is used to detect protein nucleic acid complexes, disulfide exchange that examine the mechanism of the conformational change utilizing the modified cytosine and disulfide bonds, as well as the genetic approaches by the bacterial 2 hybrid, which is used to analyze the protein DNA or protein-protein interaction. The last part is the structure alpha helical transmembrane. Figure 1 shows a 7 transmembrane alpha helix G-protein coupled receptor structure. The C terminus is intracellular while the N terminus is extracellular. Figure 2 shows 6 slices displayed through the base area of the density map. Each of the 7 transmembrane helix peaks may be seen in each of these regions. The least inclined helixes 4, 6, and 7 are grey, whereas the most slanted helixes are 4 distinct colors.
Next is protein domain of myosin. Myosins are a diverse family of proteins that exhibit conserved structural features. They consist of a motor domain, which contains an actin binding site and an ATPase activity site, responsible for generating force and movement. The tail region of myosin exhibits considerable variation, conferring specificity to different isoforms. Next is the classes of myosin. There are two classes, which is conventional and unconventional. These are the classes in each category. Next is the structure of myosins. There are 3 domains on every myosin. A catalytic motor domain with actin binding and ATPase sites is located at the N terminus of the protein. 1 to 6 IQ motif with the amino acid sequence IQ XXXR GXXXR are found in the central lever arm region that follows this domain. The C terminal section is known as the tail, and it frequently includes cargo binding domains that engage with particular adapter protein or lipids, as well as a alpha helical coiled region for dimerization. The tails of the myosins are the most hypervariable areas and have a direct role in defining the cellular functions of the various myosin classes. Next, we move on to the protein-myosin interaction. Actin, a cytoskeletal protein, serves as the track along which myosin move. The actin binding site on the motor domain of myosin interacts with actin monomers, forming a reversible and dynamic complex. This interaction is regulated by the ATPase activity of myosin, which control the affinity of myosin for actin. ATP binding leads to the dissociation of myosin from actin, while ATP hydrolysis promotes reattachment, resulting in the characteristic bulking motion of myosin along actin filaments. Next is the protein analysis of myosin. Myosin proteins are essential for the cellular movement and perform a wide range of functions within cells. Myosins also engage in interaction with various accessory proteins, include light chains, actin binding proteins, membrane proteins, and signaling proteins. Myosin isoform interact with the signaling proteins, enabling bidirectional communication between myosin-dependent contractility and intracellular signaling pathway.
Now we're moving on to the next example, which is enzyme phospholipase D1. Phospholipase D1 is an enzyme that breaks down the phosphotaglycan. This enzyme contains 3 different domains, which are FOX-PX domain, pextrin homology PH domain, and 2-phospholipase D-PLD domain. FOX homology domain is a 120 amino acid phospholipid binding domain and found in about 350 protein. It is linked to a variety of functions, and the structure of FOX domain are used to elucidate the structural basis. As for the pextrin homology PH domain, it was found 22 years ago in proteins, and it is also known to have many functions. It comes with 3D structure with 100 residue module and was disclosed by elucidating its unique 7-stranded antiviral beta sheet and C-terminal alpha helix. PLD is a transphosphatidylases that hydrolyzes the membrane lipid PC to produce the dynamic lipid second messenger phosphatidic acid PA under health condition. The structure for both PLD1 and PLD2 is similar with 57% amino acid conservation, and both have tandem PX and PH domain that are conserved. As for the protein analysis used to detect the protein is the transphosphatidylation assay, which is used to evaluate the mammalian PLD activity in vivo.
Next, we will move on to pyruvate kinase. The diagram shows the domain organization of pyruvate kinase, which consists of A, B, C, and N domain, where N domain is an extra N-terminal alpha helical domain that can be found in some eukaryotes. The active site can be found in between domains A and B. These both domains work together to create an enzyme active site and catalyze a reaction. Besides that, metal ions are also depicted in the diagram. The hinges in pyruvate kinase definitely plays an important functional purpose. For example, the hinge residues for the binding, which are located at the intersections of A and B domains. Additionally, pyruvate kinase interaction with PFK1 are responsible to maintain coordinated glycolysis control and also efficient energy output. For protein analysis of pyruvate kinase, it can be performed through STS patch or Western blotting.
In conclusion, protein domains play a critical role in understanding protein structure-function relationships and are key targets for studying protein evolution, protein engineering, and drug design. They are independently folded regions that can perform specific functions, such as binding to other molecules, catalyzing reactions, or mediating protein-protein interactions. By combining the multiple domains, proteins can achieve diverse function and participate in complex biological processes.